Okay, hi everyone. It's a good noon, everyone. It's my great pleasure to have Dr. Shilpa Garg to present her research at our CPCB seminar series. Shilpa is a tenure track assistant professor at the University of Copenhagen, Denmark. And before this, she was a NIH K99 R00 research fellow at Harvard Medical School, and she finished her PhD in computer science at Max Planck Institute of Informatics. And she is passionate to apply the power of computers to understand the biology of complex disease analysis. And she has published in ISMB, ESA, and in prestigious journals like Bioinformatics, Nature Communications, and Nature Biotechnology. And without further introduction, let's welcome Shifa to give us her talk.
Thank you, Min, and thanks everyone for joining my talk today. Good afternoon. I'm Shilpa Garg. I'm a tenure track assistant professor at University of Copenhagen in Denmark. And the topic of my talk is efficient high-resolution approaches for integrative sequence analysis of complex diseases. So here's the outline of my talk. I'll first present a fundamental biological problem, which is haplotype phasing. And I'll give a reference-based dynamic programming algorithm to solve the phasing problem. And the next, to remove any reference bias from the analysis, I will present a new workflow for performing chromosome scale haplotype resolved assembly for human genomes. Then I will give you a generalized graph-based framework of phased assembly that can be applied to any type of genomes, whether it's humans, non-humans, or clinical samples. Then I will finally present the applications of these methods to medical and population genetics. So let's get started. First, what is haplotyping and phasing? Humans are diploid, which means there are 2 homologous copies for every chromosome. Imagine you know 2 variants and you know the genotypes of these variants, AT and CT on the genome. But what you actually want are the haplotypes, which means A and T are connected on a same copy and T and C are connected on a different copy. And the process of obtaining these haplotypes is known as haplotyping or phasing. It has several applications. It can be used to study human migration, evolutionary selection, and population structures. And it also is effective to understand the causes of diseases. So the advances in sequencing technologies have created enormous opportunities to assemble these genomes and do the phasing, starting with the second generation sequencing with Illumina reads, where the reads are about a few hundred base pairs, to more recent sequencing technologies such as PacBio and Nanopore, where the reads are longer to about more than 100 kilobase pairs in length, and to more recent ones, TransSeq and 10x, where you can get a long range information on the genome. So now the goal is to use these sequencing technologies and develop new algorithms to phase the genomes. And this process is known as read-based phasing. It has several applications and advantages. The most important is it can phase de novo or any type of variations that are rare in population. Traditionally, there are 2 types of approaches used for phasing. One is the genetic, where you are given the trios in a population, and you try to predict the haplotypes of a target sample based on the Mendelian law of inheritance. But the disadvantage here is that you cannot face the variants that are heterozygous in all individuals. For example, here you see these variants are heterozygous. You cannot face this individual in this population. The next approach is statistical phasing, where you are given the reference panel from populations and you try to predict the phasing of a target sample based on the evolutionary model that is explained by the common ancestry information here. Imagine on the x-axis, you are given a reference panel of first 1,000 genome and HRC, where the number of samples are about 1,000, and the y-axis is the phasing quality. So here in this plot, you see that when you use a panel with about 1,000 samples, the phasing error rates are about 1.5%. And when you increase the sample sizes to more than 100,000, on the right, you see that the phasing error rates dramatically reduces to less than 0.5%. So this indicates that you require large sample sizes to get an accurate phasing, which is actually one of the limitations of statistical phasing. And also in statistical phasing, you can't phase de novo or rare variants, and you can't get a chromosome scale phasing, like end-to-end phasing for the whole chromosome. And you can't phase clinically relevant regions such as HLA and KIR, and I will go more in detail in HLA and KIR in later part of my talk. So to overcome these limitations, here I follow a read-based phasing, where you are given a reference genome and you are given a variant and you know the genotypes. You take the reads of the target sample and align these reads to the reference genome. So the alleles that cover these variants, they are encoded in red. And the goal of read-based phasing is to partition the reads to two sets that represents the two haplotypes. Imagine you are given a perfect case when there are no errors in sequencing data. Then you can easily make this partition such that the Hamming distance between any two pairs in any partition is 0. So for example, the alleles in these red cluster matches, and then the alleles in blue clusters also matches between any two pair of reads. But in practice, you get a lot of sequencing errors. The long reads have sequencing errors of about 10%. So the goal here is to detect these errors and flip them to get these two haplotypes. And to formulate this problem, here I followed a minimum error correction formulation, which means we flip the minimum set of entries such that a conflict-free bipartition exists, where conflict-free property holds if the alleles between any two rows in the partitions match. For example, here, if you flip this G to T and this T to G, you can make two partitions and you get two haplotypes. So next, the first question I asked was, how can we get dense and accurate whole chromosome haplotypes of human genomes? And to answer that question, I considered two latest data sets from two latest technologies. One is StrandSeq, which is actually a single cell technology, but the beauty is it can give information of haplotypes on the whole chromosome. But as you see here, there are some intervals of information, zeros and ones, and then there is no information with gaps. And then again, you see some intervals of information. So this suggests StrandSeq is highly sparse. So we combine with more local and dense information from the long reads and solved it a MEC formulation using a dynamic programming algorithm. And I implemented this algorithm in WhatsApp, which is highly optimal and efficient solution to solve phasing. And then I performed various experiments on combining different types of data sets and how the phasing is. So in these plots, each color represent a phasing segment. So when I combine PacBio with the 10x data, you see by using all of the PacBio data, you see a lot of colors in the bottom that suggest the phasing is highly fragmented. Whereas what we want is one color throughout, which means we have one haplotype through the whole chromosome. So then I combined StrandSeq with 10x data, and here we, for the first time, produced a chromosome scale phasing. As you see, there is one red color through the whole chromosome. And we also looked at the quality of this phasing. So we observed that we produce phasing error rates of less than 1% in this. So this analysis suggests that we are now in an era to produce high quality phase genomes for humans. And going back to the methodology of analyzing this genome, so here you saw that we considered a reference genome, and therefore the whole analysis is biased towards the reference genome. Imagine you take a highly diverse genome, for example, an African genome, and you would like to produce a phase genome for that. In that case, the previous methodology will fail. So to overcome that, here now, the next question I ask is, how can we get accurate chromosome scale phase genomes for humans? And to answer that, here I mainly leveraged two data types. One is PacBio Hi-Fi, which is a new technology, new chemistry from PacBio, which can produce average read lengths of about 20 kilobases, but the error rates are very low, about 1%. And in case the StrandSeq is not available for more long-range information, Hi-C is very valuable. So the next question is, how can we combine Hi-Fi and Hi-C data sets into an integrative framework to produce two haplotypes for every chromosome? So a little bit background on a genome assembly problem, which has actually started 3 decades ago with the start of Human Genome Project. So at that time, there were short reads. So the short reads, they were not able to span the repetitive regions. For example, here you see R represents repeats. And then if the reads are short, for example, here, they can't represent which repeat these reads are coming from. Therefore, when we constructed the graphs based on the short reads, where each read is a node and the connections are based on the overlaps between the sequences, so you get very tangled graphs because of the repeating structures on the human genome. But as we moved from short reads to longer reads, which has enabled to span these repetitive regions and uniquely assign the reads to repeats, the graphs look less tangled, and thus we get contiguous assemblies. But in these approaches, the main limitation was that we get collapsed haplotypes, that the two haplotypes are merged together to produce only one consensus sequence. But for human genomes, we actually want two haplotypes. So there has been some work related to diploid genome assembly. For example, Falcon Unzip and Trio Canu. The major limitations here was, one is the trio information was used which are not readily available. And the next is the trios, they lose sensitivity in the repeating regions because the underlying data used for the parents was short read data. And Falcon Unzip can produce haplotype blocks of a few megabases and can't produce the chromosome scale assembly. So to overcome these limitations, here I proposed a new workflow which starts with HiFi data, and we first produce contiguous sequences from the HiFi data, and then we scaffold those sequences using Hi-C to produce the unphased scaffolds. And now these scaffolds are whole chromosome scale. Further, we remap the HiFi reads to call the heterozygous SNPs, and we use heterozygous SNPs to basically partition the HiFi reads and the Hi-C reads to two sets, and then assemble them separately to get the phased contigs here. And we were interested to know the runtime of this workflow. So we, for the first time, can run this analysis within a day. And then I performed various experiments on public genomes to evaluate the performance of this workflow, and compared against Ryokanu method, which is the state of the art. So we observed that we produced 3 gigabases of each haplotype, which is as expected. And we can produce better continuity for our sequences compared to Ryokanu. And we could phase larger fraction of the genome as compared to Ryokanu. And therefore, our SNV sensitivity is much higher than Ryokanu. And we observed similar trend even for other genome. So this suggests that we can now produce high quality haplotype chromosome scale haplotype resolved assembly of human genomes without any reference biases within a day. So next, if we look more closely into this analysis workflow, we observe that there is an intermediate step of collapsing the haplotypes. And therefore, in case we would like to work on genomes that are highly divergent, like for example, plant species, or any clinically relevant regions which are highly polymorphic in, for example, HLA and KIR, or cancer genomes. So this workflow will have some limitations. Next, I give an introduction to a new graph-based approach that involves no collapsing to enable routine production of haplotype resolved assemblies of humans and cancer genomes. So the main idea is here to use phasing information within the assembly process. So when we look for the overlaps in the reads, additionally look for the heterozygous SNPs to find which read comes from which haplotype. So in this way, we can encode this whole phasing and assembly information into the graph, and we start to see some nice structures in the graph, which are these bubbles, which actually represents the haplotype variation. And here you see the bubbles have a common start and end node, and then you see these haplotype allele paths within the graph. In this way, you can encode the haplotype as well as any structure rearrangements in the genome through this graph. And so the whole workflow to combine HiFi and Hi-C data is as follows. First, I construct a HiFi assembly graph and then align the Hi-C reads through the node sequences in this graph. So there are two types of connections. One is Hi-C gives connections on within the components, how the sequences over several megabases should be connected to each other. And also, Hi-C gives information across components, how, for example, different arms of the chromosomes from different components, and then how these arms should be connected to each other is also an information encoded using Hi-C through these graphs. And based on essentially the maximum likelihood framework, we try to produce the phased scaffolds through these graphs. So as I said, it has several advantages that Hi-C can give information how the connections, how the chromosomes arms are connected to each other. For example, we could reliably connect the arms of chromosome 6, 1, and others. And also, Hi-C can help to disentangle the chromosomes. For example, because of this repeating structure, there are two chromosomes that got merged in one component. Because Hi-C reads of these components, they are in different territories. So we can also disentangle the different chromosomes from these using Hi-C connections. Next, I applied again this new method on a public genome, and here I observed that I can produce very high quality for the first time Q50 genomes, and also the N50s are more than 130 megabases, which suggests that these phase scaffolds are on the whole chromosome scale. And the phasing error rates are also comparable to state-of-the-art methods. Next, once we have these methods ready, what are their applications to in biology or in medicine? So the first, I more closely looked at a few clinically relevant regions, for example, HLA, which is actually a highly polymorphic regions with many variations, and it's unique to every genome and has a known associations with autoimmune, narcolepsy, and so on. So I considered haplotype sequences from 3 genomes and aligned them to the reference genome. And darker the color is, higher is the divergence of these sequences to the reference. So here you see that there are many regions which are highly diverged than the reference genome. And through our phasing methods, we could easily reconstruct these sequences. And similarly, we also looked at key regions, which is another interesting clinically relevant region, and also has tandem array of repeats. And we saw that we could also reconstruct this region reliably. Next, we looked at one PopGen gene, which is PRDM9. So in this gene, you see that there is a zinc finger array, which means there are sequences that are repeating about 1,000 times. And that happens in both humans and mouse. And on the left bottom, you see short read alignments for the reference genome. And on the top, you see our phased sequences aligned to the reference genome. So as you see, the short reads are highly confused because, again, of the repeating structure of this PRDM9, whereas our phase sequences align pretty well through them. Next, we were interested if we could also apply our method to analyzing the Neil GWAS studies. So we took one case control study where PGP1 is a case and HG002 is a control. And PGP1 has a known phenotype of narcolepsy. And we observed that when we computed a statistical significance of PGP1 to narcolepsy panel available from Neil's lab, we observed that on chromosome 19, there are about 25 variants out of 40 variants that have a high statistical significance, indicating that the variants that we found using phased sequences that also would be relevant in GWAS analysis because they have a high statistical significance to Neil's lab narcolepsy panel, whereas we didn't observe this for H002, which is a control. Next, we sequenced a cell line, which is a melanoma cell line, COLO829, and we also characterized some of the SVs using our assemblies, and we found about 26,000 SVs that were not available in short read analysis. So to conclude, in this talk, I first presented a new phasing algorithm that combines transect and long reads for doing any robust analysis of human genomes. Next, I presented a generalized approach to chromosome scale haplotype resolved assembly that without any reference bias. Finally, I gave you a graph-based method for haplotype resolved assembly in any type of complex genomes. And I presented a few applications of these methods to clinically relevant regions and cancer genomes. And I believe these methods would be helpful to the community who are interested in characterizing SVs and studying the mechanisms of diseases. And I would like to acknowledge the support of my mentors and my collaborators. Thank you.
Okay, thanks, Shilpa. So are there any questions from audience? Please feel free. Are there any? Hello, can you hear me? Hello? Yeah. Okay, thanks, Shilpa. Are there any questions from audience? Please feel free to open your mic. Yeah.
Well, I have a question. I'm wondering at some of these algorithms, how much of a problem it is dealing with the different error characteristics of some of the long read technologies, and some have much higher error rates than the short ones, or just unusual kinds of error profiles. Is that a problem for these algorithms?
Yes. So the last algorithm that I presented here is actually basically designed for the latest hi-fi data, because to look for finding the SNPs true variants from the hi-fi is slightly easier, and it's doable within the overlapping stack. Whereas if you want to work on the longer reads with 15% error, you need much higher coverages for finding the true SNPs.
Okay, are there any other questions?
So I have a question. So this is great. So how generalizable this is if people wanted to apply this to not just the diploid genome, but like aneuploid that let's say in plants or in tumor context?
Yeah, that's a good question. So this algorithm, I believe, is generalizable to plants, like a ploddy of around 4, but I have not yet tested for more than 4. But in principle, it should work. And I'm happy to take this discussion offline if you want to explore how well, if you have how well it works beyond 4. Yeah.
If I could ask one follow-up to that, I'm also curious if you have something like a cancer genome where you have a lot of local copy number variations, you might have different copy numbers at different points in the genome. How generalizable is it in that case?
So in this algorithm, we don't give a ploddy number. It's essentially, the idea is we try to leverage the variation in these ploddy to separate the reads, which aneuploidy, which haplotype the reads are coming from. So even if this structure, if there is an aneuploidy copy number change only locally, I believe we could observe those structures in the graph. But I have tested only for colo 829, which is a trisomy, like there is a 3 change, but it's a good question that how further it can go in practice. But in principle, I believe it should work.
Okay, thank you.
Are there any other questions? Okay, so I have a small question. So do you have any comments about the computational cost of these algorithms and the scalability?
Yes, so to make them work well in practice, so generally I have used some greedy heuristics. So, but these phasing, which is on the reference genome, is like to power the coverage into the number of variants on the reference genome, because it's just linear. But on these phased assembly graph, it's because we align all reads to all, so there is all versus all alignment, so there is a quadratic runtime. But by using some sort of greedy heuristics and also using the domain knowledge, I think I have made them to work in practice.
Okay, thank you. Yeah. Are there any other questions from the audience? Please feel free to open your mic. Okay, so if no further questions, so let's thank again Dr. Garg for her wonderful talk. So this concludes today's seminar.
Thank you, everyone.
Thank you.